Today is Lab 12, just one exercise. We're going to do bacterial transformation using the PGLO system. So this is a kit that we purchased from Bio-Rad, one of the biotech firms. So transformation is a process in which you're taking DNA and putting it into an organism. And because of that, DNAs remember information, and they can use this information and actually make something and do something. In this case, one of the genes, one of the DNA segments we're giving them is the ability to make green fluorescent protein. And one of the nice things about this genetic change is that it produces a product that you can see, right? So you can actually see that they have this genetic information because they will fluoresce under UV light. They take the energy from the light and they use that to fluoresce, to bounce back another light or color. So in the case of this picture here, we're going to do it for bacteria, which are what cell type? Prokaryotes or eukaryotes? Prokaryotes, where mice, and like ourselves, are eukaryotic. So this can be done for, higher life forms, you could say. It's done a lot in bacteria, but it's also done in research for eukaryotes as well. And so as you can see under the UV light, two of these mice are glowing, right, because they're expressing that green fluorescent protein. Now they didn't just do this because it looks cool, right? They did this because they wanted to know, they're kind of using it as actually a marker, because in addition to the green fluorescent protein, they probably gave it other genetic information. And what they're doing here is being able to very easily visualize whether they've taken up that genetic information by the fact that they're expressing it and glowing green. So whatever else they tried to change about these mice was successful. This isn't always successful. So notice the mouse in the middle is not glowing, right? He's not been transformed. He didn't take up the DNA. It didn't get utilized for some reason. So of course that one they would not use in their experiment because it's not been genetically altered. Does that make sense? Because can you see DNA? No. You wouldn't know if somebody's DNA is different unless you literally tested it. So instead of having to run the test on each one of these mice, you can very quickly shine a UV light on them and go, okay, that one, it worked. The other ones did not work. Make sense? So one of my favorite experiments is one where visualization was very important and being able to tell genetically one organism from another. So I show this to my kids that I taught this summer to. Whenever you say the word sperm, they pay attention, right? Even happens for my college students. So fruit flies mate with more than one male at a time. So the hope is, and the belief is, that then her offspring will be better suited to the environment because they're genetically diverse, right? Because she mated with more than one male. They're not monogamous. So they wanted to see what is happening in the female reproductive tract. So she'll mate with several flies, male flies, and the sperm in the reproductive tract, they've actually been able to see that they compete with one another. And the reason why they've been able to see this is because they genetically altered the sperm of one of the fruit flies, the male fruit flies, to be red and the other one to be green. So when she mated with both of these flies, you can see that competition. They were able to visualize this by making those sperm glow the two different fluorescent colors. You could tell one fruit fly's sperm from the other. So this is a short video of that interaction. And the red ones are kind of hard to see. So you got to kind of focus. And the green are very bright, right? And you can see them swimming around in the reproductive tract. Like I said, it's short. I'll play it one more time. And the goal is to get to the egg, right? Or eggs in this case. So the other thing that they found is they did additional testing, and they were able to find that actually the female reproductive tract produces chemicals that tries to stop this competition. Because again, she wants her eggs to be fertilized by multiple different sperm from different fruit flies. to have a diverse population. So it was very interesting. I mean, they wouldn't have been able to see that unless we were able to genetically alter those sperm and actually see the competition that happens between them. Click the wrong thing. I want to click over there. Did I open up more than one of these? Possibly I did. So this, on the other hand, is a drawing on a petri dish that people did with genetically altered bacteria, right? So each one of the bacteria, there's lots of different fluorescent proteins out there, not just green fluorescence. So some of the kids this summer actually worked with organisms and made lots of different colors and actually did drawings on petri dishes. Not quite to this level, they didn't have as many colors, but it was kind of fun, right? So microbiology can be art too. This was somebody very talented, for sure, and access to a lot of different strains of bacteria with the different colors. I don't know who did that. We stole that picture from the internet. But it's possible. It's actually possible to do that. So again, what is transformation? Transformation is genetic information from one bacteria to another. And what will happen is that the bacteria takes up the DNA, and they actually only take it up single-stranded. And then they'll find a place within their chromosome, usually to incorporate it, and then they'll make the complementary strand. So when cells die in the environment, their DNA becomes part of the environment. So that what we would refer to as the donor cell, right, has died, and its information has become part of the environment. Other cells, probably as a way of DNA repair, have gained this ability to be able to take up DNA from the environment and utilize it if they choose to do so. In that process, they become what we refer to as transformed, because they're genetically different now. And they have different abilities because of that genetic information they have picked up. So the analogy that I always use in my lecture classes is transformers, right? So transformation, transformers, same thing, right? So in the case of transformers, they're cars that can turn into robots, right? They can make that transformation. Of course, that's fictional. Still trying to explain that to my son. There's someone who drives a Camaro around New Orleans. We see it occasionally. Thinks it's going to turn into Bumblebee. Said, nope. But, who knows? Someday it could really happen, right? The amazing things that have happened. But at this point, it's not going to transform into a robot called Bumblebee. But we can make bacteria, eukaryotic organisms, as I said, we can give them genetic information. And so one of my favorite examples, too, is that human insulin is actually made by bacteria. We've given them the genetic information. Of course, it's done in a lab. This is biotechnology at its best. And it actually makes it exactly like humans would. And so this is able to manufacture it very quickly and cheaply for individuals that are insulin-dependent diabetics. The other thing is they've gone one step further, and this is a protein that isn't very stable under high temperatures. Usually has to be kept refrigerated. And so they've done some changes, some tweaking to the structure of that protein that makes it more stable at higher temperatures. So they can keep it at room temperature and not have to refrigerate it, which is really beneficial. And it still works and is still recognized by the body as human insulin. So some pretty amazing stuff that can happen. And then, of course, people know about genetically modified foods, right? And there's debate on whether that's good or bad, right? I have to say I don't know what side of the fence I'm on that. Jury's still out on me. I'm still gathering information on how I feel about that. So, why do we talk about transformation? Well, transformation has been happening for a very long time. Like I said, this ability of bacteria specifically being able to pick up DNA from the environment. When they do this, they pick up information and potentially on how to do things. And so this has led to what we refer to as superbugs. So these are bacteria that have become resistant to antibiotics, right? So they're not good news for us, definitely good news for them. So back in 1990, a Swedish man acquired a Klebsiella pneumoniae infection while in India that was resistant to a particular class of antibiotics similar to penicillin and ampicillin. And Klebsiella pneumoniae is a gram-negative, right? We've worked with this. Can cause a really serious form of pneumonia. It's encapsulated, which helps protect it. So this drug is very similar to ampicillin, penicillin, what we call beta-lactam drugs. They have this ring structure in here. And come to find out, what was going on here is that the Klebsiella pneumoniae had acquired a plasmid. And this is a circular piece of DNA in addition to their chromosome. And that's what we're going to be working with today. We're going to be actually working with a plasmid, a circular piece of DNA that we're going to transform our bacteria with. So they're going to pick up that whole big circle, well, it's not big, but relatively small circle of DNA, which is information. And in this case, they found that on that plasmid, there was a gene that they have abbreviated BLA, which stands for beta-lactamase, and this is an enzyme that breaks that beta-lactam ring. Of course, the ND stands for New Delhi, and I guess that's probably maybe even the Providence or something like that. So similar strains of Klebsiella pneumoniae contain these enzymes, right, that can destroy this particular group of antibiotics. This gene, they believe, has been picked up by other bacteria, right? We see this gene a lot, unfortunately, and so it's created quite serious problem in that we have a lot of bacteria that have gained this enzyme that can destroy penicillin-like drugs. So as of 2010, they have found 180 isolates, different bacteria that have this gene, right, that can destroy beta-galactanase drugs. So the other thing is that one of the drugs that's commonly used, known as Augmentin, it is a penicillin-like drug. I want to say it's amoxicillin. Don't quote me on it. And a drug. And what the drug does is it inactivates beta-lactamase enzymes. So that way that penicillin-like drug, amoxicillin, can still work. It's not destroyed if the organism has this enzyme. So that's one way, right, we can kind of combat this problem. Or we give different types of drugs, right? Different drugs that aren't affected by the beta-galactanases. The problem with some of these, right, is that they're not as effective, right? And especially with colistin, it can be toxic. By a similar mechanism, Mectillin-resistant Staphylococcus aureus, you've probably heard of, we've mentioned it previously, right? MRSA. It is resistant again to beta-lactam drugs, penicillin-like drugs. Mecticillin was the most commonly given. So they've, and I'm not sure if their resistance is the enzyme or not. If that's the source of their resistance, it probably is. And then, we run into problems with tuberculosis, where we have strains now that are referred to as extremely drug resistant. Most of the antibiotics that we use against tuberculosis cannot be used against that strain if the individual has that. So we're very limited to the amount of drugs that we can use to treat someone with a highly resistant strain of tuberculosis. So E. coli 0157. This is one you've probably heard of sometimes in the news. It has acquired the Shiga toxin. And again, this toxin is from Shigella. It's a similar bacteria to Salmonella, causes a similar type infection, really serious infection. And the bad news with this toxin is it deteriorates and attacks your intestines. It actually kills the cells of your intestines. So it causes really severe bloody diarrhea, what we refer to as dysentery. So they believe, again, that this genetic information was transferred from one bacteria from another, right? It's the exact same toxin that Shigella has, that this particular strain of E. coli has, and makes it highly pathogenic. And again, a small circle of DNA was passed from one bacteria to another. Now, how that passed, whether they picked up the DNA from the environment or the two cells joined and transferred that via conjugation, or whether it was infected by a virus and it picked it up and transferred it, probably not the case. I would bet conjugation was probably how this DNA got from one cell to another, what's sometimes referred to as bacterial sex, because they literally join up and exchange information. Virus can infect bacteria. They can pick up DNA and transfer it to another bacteria. That's called transduction. What we're doing today is we're just putting DNA into the environment, so we're doing transformation, where they're going to pick it up and they're going to transform, they're going to change based on that genetic information. They're going to get new abilities. So natural competence, only about 1% of the known bacteria are naturally what we refer to as being competent. This means that they have the ability to pick up the DNA from the environment. Transformation in nature is very complex developmental process. There are several genetic information that's necessary, and they found that there's 40 different genes in Bacillus subtilis that allows it to be able to do transformation, to be able to pick up DNA from the environment and incorporate it into its chromosome. This requires energy, right, to actually pick up DNA from the environment, bring it in. But it has evolved, we believe, as a way of DNA repair. The problem for prokaryotes like bacteria is they usually only have one copy of their genetic information. They're what we refer to as haploid. We, on the other hand, are what? We're diploid, right? Because we have how many copies of our genetic information? 2. One you got from mom, the other you got from dad, right? So you're the combination of both. You actually have 2 copies of most things. So if mom or dad gave you a defective copy, hopefully the other one gave you a good copy. You end up with genetic disease when you get 2 defective copies, right? You end up with things like sickle cell anemia, where you can't properly make your hemoglobin because neither one of your parents gave you the proper information on how to do that, right? So when these guys go get damaged genetically, they've got to be able to repair that, and one of the ways might be to pick up DNA from the environment and maybe able to repair what got damaged, and maybe be able to get something that's beneficial, right? Especially when it comes to antibiotic resistance, that's going to make them more advantageous for the environment. So they're going to do this under stressful conditions, and again when they've been damaged, if they have the ability to do it. Some people refer to it even as a primitive form of sexual reproduction, because again, all sexual reproduction is exchange of DNA, right? All sperm are mobile DNA, right? But you need that DNA, right, to make another human being. For bacteria, do they have to have genetic information from another organism? No. They just replicate by binary fission. You can basically say they just have moms, right? And they're an exact copy of their mom unless their mom made a mistake in that copying process. So they don't need another organism, another bacteria to donate genetic information. But it can happen, right? So you could say it's a primitive form of sexual reproduction. So we can cause this to happen in the lab. One of the ways we can artificially cause cells to be competent, to want to actually take in DNA from the environment, is using electroporation, where we basically shoot it with an electronic field. This, of course, makes the membrane permeable and allows the DNA to come into the cell. The cells we can cause them to take up chemicals and drugs and DNA, right? So not just DNA, we can get them to take up other things from the environment in this process. The technique can be applied to many cell types, not just prokaryotes, right? They can do this with eukaryotic cells as well. It's costly though, right? It takes a special machine that's going to apply the electronic field, special cuvettes that you put your samples in. So needless to say, we don't do that here at Delgado, right? We're not doing transformation, but who knows, in the biotech, they may advance to doing electroporation. They've got some pretty high-tech stuff. And remember, they're having the open house Thursday night, so if you guys are interested, we'll be here Thursday night from 5 to 7. So chemically competent, and that's what we're going to be dealing with today. We're using calcium chloride solution, what we call a transformation solution. And these cations help by creating a field that allows, remember, DNA is negatively charged, the negatively charged DNA to permeate through that membrane. And it actually will even allow the double-stranded, where normal transformation is just going to be single-stranded. That will be taken up. Well, I said that wrong. Double-stranded binds, but the single strand still just goes in, right? A single strand of it. But we're not going to have to worry about it finding the homologous part. It's going to stay as a circular plasmid. It's not going to incorporate into the chromosome. So again, you can buy kits, commercially available kits, that allow you to do transformation. This is from Invitogen. The one we buy from Biorad is specially designed for in the classroom use. But you can buy ones that have different levels of efficiency, how much transformation happens, how very competent the cells are. So depending on what type of experimental situations you're doing. So they provide you everything you need, including the media, positive control, aliquots of the bacteria itself to be able to do your transformation. So I worked in a lab when I was in grad school where I had to do some transformation. And it was mostly for getting the bacteria to copy the plasmid for me, so then I could lyse the cells and get out the plasmids, and then I sequenced that genetic information. So I was able to put DNA from frogs into bacteria, get them to copy it for me, so that then I could run it on the sequencing machine and find out the sequence of the DNA. So this type of work is done all the time, especially in genetics labs. And, you know, this stuff would be kept in the freezer, and I would run through the very similar process that you guys are going to do today with my samples. But I did a whole bunch more than you guys did. So bacterial transformation, we said this is a process in which they're taking exogenous, outside of their cell, DNA and bringing it in and incorporating it into their genome. Only competent cells have the ability to do this. So it means they have the ability to take up DNA. One of the properties that we're going to be dealing with today is bioluminescence, right? And this is the production or the emission of light by a living organism as the result of a chemical reaction during which the chemical energy is converted into light energy. We're dealing mostly with emission, where they're going to be absorbing the UV light and emitting fluorescent green light. This ability and the gene that we're using comes originally from jellyfish. So jellyfish are some of the aquatic animals that have the ability to fluoresce. So if you've ever been to the Aquarium of the Americas, you'll notice that the jellyfish are in relatively dark tanks. This is to help in the visualization of them. I'm not sure any of the ones that they have there, if they have fluorescent abilities, particular ones. There's one that's one of my favorite ones that's really pretty. If you actually read the placard that's next to it, when it's moving, it looks like there's rainbows running along it. And what it is, it has tiny little projections like cilia. And as they move, they refract the light. So it's like little prisms running all along their little bodies. It looks like they have shiny rainbows running along them as they move. They're really pretty. Different from bioluminescence, though. So, again, they're absorbing the UV light when we shine the UV light on them, and they're going to bounce back that light in fluorescent form. So electrons actually get excited when we shine the UV light on it, and then that movement of those electrons back to their ground state or close to it is what generates the fluorescent light that we will see, the fluorescent colored light. So Peter's a big fan of explaining all the math. Me, not so much. So the slide is mostly for him. But the take-home message is wavelength, smaller wavelengths means higher energy. And so notice that UV light is a much smaller wavelength. It's a higher energy light. And the visible light spectrum that we deal with, a little bit larger wavelengths, not as much energy, but again, we can visualize it. UV light you cannot see, but it's much more high energy because it is a much smaller wavelength, right? About 1 nanometer for each wave, where when you're in the visible light spectrum, right, we're only talking about 700 to 400 nanometers. Much larger wavelengths, less energy. Still energy, right? Not as much. So all the conditions have been worked out. Like I said, this is a kit, right? It works just about every time as long as all the pieces are in play. So we know how many cells to use. We know how much DNA to use. We know the transformation efficiency, how well this works. So how many transformed cells we'll get per milligram of DNA that we'll add to our sample. And we're going to spread a set amount onto our plates. So just like when we did dilution, right, and plating and counting with that, you could essentially go back and count how many cells you have that transformed on that plate and figure out the efficiency. So we should get about 75 transformants, so 75 little colonies on each plate by plating 100 microliters of our solution. So everything is measured out for this experiment so that it works. If you didn't know, you would make serial dilutions, right? You would dilute out and do several platings to figure out what the efficiency is. So the plasmid itself that we're utilizing today, as I said, is a specially designed plasmid known as the PGLO plasmid. P for plasmid, GLOW for the fact that it allows them to glow. So some of the genes that are on this plasmid are, of course, the BLA that we've spoke about. This is the beta-galactanase enzyme. And we're going to be using the antibiotic ampicillin, which is a penicillin-like drug. So again, it has that beta-lactam ring. So giving them this enzyme, they're going to be able to destroy ampicillin. So this is a selective pressure for our transformants. Because what's going to happen to the E. coli that doesn't transform in the presence of ampicillin? It's going to what? It's going to die, right? We saw this before on our Kirby-Bauer plates. Ampicillin kills E. coli, right? It's one of those modified penicillins that actually works on gram-negatives like E. coli. So we're going to give it this resistance. And this is going to encourage them to keep the plasmid because we're going to grow them on plates containing ampicillin. So if they don't keep it, they will die. So this is what we refer to as constituently expressed. So as soon as they get this plasmid, they can start making this enzyme from this DNA, from this genetic information. Another one that they're going to make is a regulator protein that's important for the regulation we actually have the green fluorescent protein under. So that gene product right here, ARAC, again is constituently expressed, means as soon as we give them the DNA, they can make this protein. This protein will actually bind to the plasmid, and it's going to regulate the expression of the green fluorescent protein. OREI is the replication site, because once this is taken in, bacteria will actually make multiple copies of their plasmid before they're even getting ready to divide. Why they do this, we don't quite know. I guess to be able to, if the more information you have, the faster you can utilize it and make products. It's the only thing I could think of that would make sense. And then, as I said, this green fluorescent protein we actually originally got from jellyfish. And that's the actual species of jellyfish that it came from. So as I said, constituently expressed means it's not under regulation. They're gonna make it all the time. It takes energy to copy this plasmid to utilize this information. So again, giving it antibiotic resistance, and putting it in an antibiotic environment is going to encourage them to keep it. And also, if they don't keep it, they're going to die. So we can really select for only the ones that have transformed by applying that selective pressure of ampicillin. So we're not doing it to make super bugs, right? We're doing it to try and make sure they stay transformed. So notice I mentioned regulation. Some of the genes that we have are under regulation. We're only going to utilize that information unless we need it. So the same is true for things like bacteria. They don't always utilize the information they have, especially when it comes to sugar metabolism. For instance, E. coli, another operating system or operon that's commonly used is what's called the lac operon. That's the one for the digestion of lactose. They don't utilize the enzymes for the digestion of lactose. They don't make those enzymes all the time. They only make them in the presence of lactose. So when lactose is available, they make the enzymes to digest it. They have that genetic information, but they're not always expressing it. So the same is true in our own cells, right? Our red blood cells make hemoglobin like crazy, right? Because that's part of their functionality of what they need to do. Your liver cells, on the other hand, do they need to make tons of hemoglobin? No, it's not their job. Do they have that DNA in their cells? Absolutely. It's just not turned on. They're just not utilizing it. They're just not making that protein because they're not being signaled to do such. So the environment of our body is dictating what cells do what. The environment in general will dictate what genetic information a bacteria uses. So a lot of the DNA is under a control system. This control system is referred to as operons. It's a sequence of genetic information that functions in a coordinated manner. You have an operator, you have a promoter, and two or more structural genes. So these actually make something. So make the enzyme, say, to digest the sugars, or the proteins needed in the membrane to transport that sugar into the cell at a higher rate. So the operator is the segment of DNA to which the regulator binds. So there's some protein that's going to bind here and determine whether the information is going to be utilized or not. So inducers usually bind to the regulator, change it in some way I'm trying to, there's two different ways, and I hate the terminology that it goes. So repressors are things when they bind, they stop the information from being utilized. Inducers, on the other hand, when they activators, when they bind, they increase the utilization of that information. So we can go either way. We can repress or we can activate. And inducers are going to induce or cause it to happen. So in this case, the regulation system we're dealing with is a repressor. And the sugar itself, the arabinose, is going to be the inducer. This will probably make more sense when we get to the pictures. So that's the operator, right? It determines whether things get turned, you could say turned on or turned off. Does that make sense? A little bit? Okay. The promoter site is the actual region of DNA. Each gene set has this, and this is where RNA polymerase binds. So it knows this is where I start. And that enzyme binds to the DNA and it makes RNA, right? RNA polymerase makes RNA. But it's got to be able to bind to that promoter site in order to be able to copy that DNA into RNA. What this system does is it usually blocks the promoter site or makes the promoter site available depending on whether you're talking about repression or activation. So as I said, RNA polymerase is the enzyme that actually makes the RNA from the DNA template. So repressors are often repress and prevent, as I said, the RNA polymerase from binding, where inducers often relieve this repression and sometimes even enhance the binding of RNA polymerase. The structural genes, as I said, are the amino acid sequence to make a protein, whether it be an enzyme to digest a sugar or maybe a protein that goes into the membrane to help bring in that sugar. Instead of putting enzymes to digest sugars, we're using a sugar regulator system, a sugar operon known as the arabinose one. Instead of putting the structural genes to digest arabinose, that sugar, instead we stuck green fluorescent protein gene there, or they did, in their plasmid. And so what that genetic information allows them to do is make a protein that when hit with UV light is going to emit green fluorescence. So arabinose is a sugar commonly found in the pectin of plants. Remember, one of the constitutively expressed genes is for a protein. That protein product binds here at the regulator. It is the repressor. So it makes the promoter site inaccessible. When the effector, right, the very small arabinose sugar binds to that protein, it stops that protein from blocking the promoter site. So now RNA polymerase can bind here, and it can copy this information, these structural genes. So in this case, right, for sugar digestion. In the case of our operon, it's been changed. Instead of enzymes to digest the sugar, we're tricking it into making green fluorescent protein. So same system, right? You have the regulator protein that's produced and it attaches to the operator site. Because of that, the promoter isn't available. RNA polymerase can't bind unless arabinose, our effector, binds to the repressor. Then RNA polymerase can bind, and then the cell will make green fluorescent protein. So, under what condition will they make green fluorescent protein? What do they have to have? They got to have arabinose, right? We got to give them arabinose so it will bind to the regulator, allow the promoter to be available, RNA polymerase to bind, and they'll actually make green fluorescent protein. So only one of our plates will glow tomorrow. Right, I got some that we made last week. You guys can take a peek at some glowing ahead of time. Right? But it's only the one that contains arabinose. Right? Only one of the plates contains arabinose, so only one of them is going to glow. So this is a repressor, right, because it's stopping its ability. So for hypotheses, right, one thing cannot happen without another. So we can't get green fluorescence unless we've actually got expression, right? So if we see green fluorescence, we know that expression happened of that genetic information, that they have it. Not only do they have it, that they utilized it. So we can measure qualitatively, we can quantitatively, we can count, right? Or at least we can detect qualitative, we can look and see green, right? Fluorescence. So we know under what experimental conditions we'll be able to observe fluorescence, right? And that's when we have the arabinose. So BLA, that gene stands for beta-galactanase enzyme, and again, it can destroy penicillin-like drugs. So in the case of ampicillin, right, it gets destroyed by this enzyme. So if we give them this ability, they'll destroy ampicillin. Ampicillin will not kill them. So before transformation, can E. coli produce does it have the beta-lactamase enzyme? Does it have the genetic information to make that enzyme? No. After transformation? Yes, because we're going to give it to them. So, for today, already done for you, are 2 tubes in the kit you'll be provided with, labeled plus and minus. Each one of them contains 250 microliters of transformation solution, this calcium chloride solution. The plus and the minus mean that the plus tube we're going to add DNA to, the minus tube we are not going to add DNA to it. Why aren't we going to add DNA to both tubes? The negative one is our control. Yes. So we're going to use ice buckets filled with water. And we're going to place our white racks that you see there in the back. These actually will float. We're not going to use the foam racks that come in your kit. Those I just used to hold the tubes in your kits, so they're not floating around in your boxes. Using a sterile loop, and we're going to use the plastic loops provided in your kits. You're just going to take a little bit of bacteria off the plate that I'm giving you, and you're going to add it to both of your tubes. At this point, even though they're labeled plus and minus, they're just transformation solution, right? So you can use the same loop to add bacteria to both tubes. Ideally, if we wanted to be really specific, right, we would add it to one tube and then divide it in half. We're not going to do that additional step today. It's already been divided, the solution, so you're just going to add a little bit of bacteria to both of the tubes. At this point, neither one of them is different. Then I'm going to come around and I'm going to add the DNA to your plus DNA tube, so leave that one open in your racks. I'm also going to shine the UV light on the solution so you can see that it is just DNA. There's no protein in there, because if there was protein, it would glow, right? So this is to show you it's not a Chris Angel or David Copperfield magic trick. We're doing real science. Right? It is just DNA in that tube. So I'm actually going to use a micropipetter, and you guys are going to use some of the micropipetters today. So the loops that we use actually are calibrated to 10 microliters, but it's really hard to pick up the solution, that small amount. It's much easier to use a micropipetter, so I'm going to do that today. But again, it's a measured amount. We're going to add 10 microliters into the plus only tube of the plasmid of the DNA. Oops. I didn't ask you the question. What could I add to the negative tube? I'm not going to add anything to it because it's a really small volume. But what could I add to keep it consistent volume-wise? Just plain old water, right? Sterile water. Again, I'm not going to do that step. It's a really small amount. Doesn't affect it. So, presence of genes that lead to ampicillin resistance and green fluorescence. So remember, the BLA gene is going to give it resistance to the ampicillin. So before transformation, does it have it? No. After, will it? Yes, because it definitely won't even survive without it. Do they have green fluorescent protein gene before transformation? No. E. coli does not glow green under UV light normally. Okay, so after transformation, will they have that gene? Yes, they will. So our next step is we're going to leave those tubes on ice for 10 minutes. Once those 10 minutes are up, in your book it says to label your plates. Your plates are already labeled because it's easier for me when I'm preparing these plates, because some of them contain ampicillin, some contain ampicillin and arabinose. Some of them don't have any of those additives. It's just easier to label them as I make them to keep them straight. So you guys don't have to worry about labeling them. It's already done for you. You're going to bring your ice bucket bath to the water bath that you see in the back there that's turned on next to the racks and the refrigerator in the back. This is set at 42 degrees Celsius. You're going to pick up those little white racks that you see back there and take it right from your ice and put it into the water bath. You're only going to leave it in there for 50 seconds. So it's very important to not walk away. 50 seconds goes by very fast when you walk away. Trust me, I know this, right? Remember I said I did a lot of these, right? And I would try sometimes, right, when we're doing multiple things, and you would walk away and it wouldn't work if you left it in there too long. So the other thing is, keep your ice bucket right there too, because as soon as those 50 seconds are done, you're going to transfer your rack right back to your ice bucket and then bring your ice bucket back to your lab bench. You're going to leave it on ice for 2 minutes. Why are we doing this? Why are we going from ice to hot water to ice again? Yeah, it's going to help with the permeability, and it's a process we call heat shock. And it's basically a stressor. Because remember, in stressful conditions and environments, this is when they are encouraged to take up DNA. So this is not all that different from if you've ever gone on vacation and gone from the swimming pool to the hot tub back to the swimming pool. You know how shocking that is on your system? That's what we're going to be doing to our bacteria today. Swimming pool, hot tub, swimming pool. So as I said, the time matters, right? We don't want to leave them in too long. For this particular system, it's been worked out. 50 seconds is optimal, right? As you leave it longer, the efficiency drops dramatically. The longer you leave it under the heat condition. After the 2 minutes, you're going to remove your rack and just place it right onto your desktop. So now it's going to be at room temperature, your tubes. We're not going to use the plastic pipettes in your boxes. We're actually going to use, I've sterilized some tips. We're going to use the pipette men, and I'm going to give you a little tube containing broth food for your bacteria, and you're going to feed them. And it's already preset at 250 microliters. You can use the same tip as long as you don't stick it down into the fluid of each one of your samples. You'll be able to draw up 250, put it into your minus tube, put it into your plus tube. And then we're going to let them sit. So again, you don't have, in that case, you wouldn't have to use but one pipette, right, as long as you don't stick it down into the fluid. And same thing with the tip. So the alternative is what we're going to do today. We're going to use the pipette. Because as you can see, and as you guys have experienced, these little plastic pipettes aren't fun to work with, right? 1 mil is tough enough. Can you imagine having to draw up to this little ridge? Yeah, and then later on to that one. So we're going to use the much accurate, easier to use pipetters today. So why were the vials incubated? Why are we going to then incubate them for 10 minutes after we give them the LB broth, before we put them on the plates? Not necessarily grow, but we just gave them DNA, right? That's information. Can they use it? It's information. If I gave you the answers to the test, you'd want them. Right? Okay, so we give them this information. That plasmid, what information does it have on it? What genetic information do we give them? The BLA, right? The beta-galactanase enzyme. Are they going to make it? Yeah, they better, because guess what? Some of those plates we have contain ampicillin. If they don't make it, what's going to happen to them when we put them on that plate? They're going to die. Right? So in this time frame, they're going to make the beta-galactanase enzyme so that they'll be able to survive in the presence of ampicillin. Remember, that one's not under control. It constitutively is expressed. Are they going to make green fluorescent protein, though? No, because remember, that one is under the control of the operator. What has to be present in order for them to make that protein? Arabinose. And there's no arabinose in this broth that I'm going to give you. It's just straight up food. No special sugar. What's the other one they are going to make, though? Here, I'll rewind. I'll make it easier on you guys. Let's look at the plasmid. There you go. So we already said they're going to make BLA. We already said they can't make green fluorescent protein. This one is an abbreviation, right? This is for the repressor. That is going to be made because it's going to bind to the operator, and that's definitely what's going to stop them too for making green fluorescent protein. They're only going to make it when we add the arabinose. So now I'm going to find where I was. There we go. So definitely got to make that ampicillin resistance, right? That beta-galactanase. So now the next step, you're going to transfer 100 microliters of the sample from the DNA tube to your 2 DNA plates, plus plates. So, I don't have a lot of fancy pipetters for us today, but those little blue pipetters you see in your racks are actually ones you'll use again next week for one of the experiments we're going to use, and they're preset pipetters to 50 microliters. So if we need 100 microliters, we've got to do this twice, right? So you just take 50 microliters twice from your tube and put onto your plates to get your total of 100. For both your DNA plus plates, you can take from the same tube, you can use the same tip, right? But then when you're doing your minus plates, you got to change tips, right? Because we don't want to cross-contaminate. And so you're going to do your plus, then your minus. Then you're going to use, someone else in the group can use a sterile loop. And again, you can use the same loop for both plus plates to spread out that liquid. We're not going to use spreaders like we did before, the glass spreaders, because you're actually, the Petri dishes you guys are going to get today are small. They're only about this big. So just a loop from the sterile loop packet that you'll get, just spread it out on the surface of the plate, so again we get cells all spread out. You can use the same loop for the plus plates, and then use a different loop, right, for the 2 minus plates. Don't tape your plates together like it tells you to in your protocol, but I do want you guys to write your table numbers so that I know which tables to put them back out. I don't know if you guys noticed, but your tables are numbered. Right? So if you guys are 4, I think you're 3. I think you guys are 2. And then we'll stick them in the incubator, and by tomorrow we'll have glowing plates. So what information is provided by the LB, which means it just has food, DNA negative plates? Remember, why are we doing no DNA? What is this for our experiment today? It's a control. And this is a control so that you know that the E. coli that we started with is viable, that the heat shock didn't kill them, right? They're capable of growing on this without ampicillin. Where on the other hand, what information is going to be provided by the LB ampicillin DNA negative plate? So they didn't get the DNA. This plate contains ampicillin, so what's going to happen to these guys? They're going to die, right? And this is to confirm that the E. coli we started with was not resistant to ampicillin. Does that make sense to you guys? So my ampicillin's working too. I'm happy about that. It's usually an issue. Successfully killed bacteria with ampicillin this week. This always makes me happy. So obviously transformation could only occur if the PGLO plasmid is introduced into the solution. So which plates are going to exhibit transformation? Our DNA positive plates. So our LB ampicillin DNA positive plate will have, it'll grow. The one that has ampicillin and arabinose, it will grow as well. Only transformants. So only the cells that have taken up the DNA and utilized it, produce the beta-galactanase enzyme, are going to grow on these plates. And they can only do that, shown by our control plates, if they have the plasmid. But why is our arabinose plate gonna glow and not our ampicillin plate? Go ahead. Why is the arabinose plate gonna glow? It's going to bind to the repressor that's binding to the operator. Promoter becomes available. RNA polymerase is going to copy it. So remember, this is an inducer. Arabinose is necessary for this operon that we have this controlled expression that green fluorescent protein is under. So only one of our plates, as I said, is going to glow, because only one of our plates is going to contain the arabinose. Both of them are going to contain transformed cells. But remember, this particular one is under control. So only the arabinose plate is going to actually glow. Turn on that operon. Express that information and make that protein. So I mentioned this earlier, how efficient this system is determined the number of transformants we get per milligrams of DNA that we add to the sample. So it's independent of experimental variables such as the volume of the transformation reaction. I know this once because I made a mistake and put 500 microliters in each one of the tubes. I actually told someone else to do it for me and forgot that we had changed it to aliquoting for you guys. But it still worked, right? So volume doesn't matter. And I've done this, so I know it definitely doesn't matter. The portion of the reaction mix that's plated, or even the amount of DNA used, doesn't matter. Okay? Instead, it reflects the bacteria's competence, right? How able the bacteria that we're working with are to pick up the DNA. Okay? That's what we mean by efficiency. So it depends on the organism itself, right? And its tendency under this treatment to actually pick up the DNA. Does that make sense to you guys? So again, you can buy ones that are highly efficient. And again, it has to do with the bacteria you're using, not any of the other parameters to the efficiency. So suppose you count an average of 60 colonies per plate on 2 plates with transformants. The 2 plates containing both PGLO DNA and ampicillin, right, are transformed plates. According to Biorad, we're going to be using 0.03 micrograms per microliter. And again, that 10 microliters of that solution is what we're going to add. So I think it's what that ends up being. I'm not even going to attempt to do the math. Okay. Remember, we're going to plate 100 microliters of a 510 microliter solution. Because remember, we're going to start out with 250 microliters of transformation solution. I'm going to add 10 microliters of DNA to it. And you're going to add 250 microliters of the LB, the food. So we're going to end up with 510. So what's the transformation efficiency? First, we got to calculate, remember, take into account the volume. So if we have 60 CFUs, 60 colonies on that plate, and we only plated 1/10 of a mil, right? We have 600 CFUs per milliliter. Second calculation, the DNA concentration of the reaction mix. So again, 10 microliters. See, I didn't have to do the math, but I was going to do it right. Times by 0.03, right, gives us 0.3 micrograms of DNA we're adding into that tube. So 0.3 divided by 0.510, right, because that was microliters, we want to put it in milliliters, ends up being about half a microgram per milliliter. So now we have 600 CFUs per milliliter divided by the concentration of the DNA, gives us about 1,200 CFUs per microgram. So this pretty good, right? And in fact, their system, they say, they range between 800 and 7,000. I think that's a pretty big range, right? But we're definitely within that range if that's the type of results that we get. So again, as I said, on average, and I haven't counted, but if anyone feels like counting today, I've got plates. We could count, right, how many colonies they ended up with on their transformation plates and see how efficient it was this last go around. And the same holds true when you guys get your plates next week. So again, we're going to have 4 plates. They're already labeled as you see here. Plate A, right, is going to be our DNA negative plate. Just LB shows us control. So we're going to get a complete lawn of growth. There's no reason why they won't grow on these plates. Unlike the DNA negative ampicillin plate, are we going to have any growth on this plate? Shouldn't see any growth, because remember, plain old E. coli that we start with today, well, it's not plain old, but E. coli should be killed by ampicillin, right? Otherwise, we can't select for transformants on our ampicillin plates. So our 2 other plates contain ampicillin, right? They're going to grow, hopefully, only transformants. So you'll see individual colonies. Like I said, if we spread them out good, we can even count them. But if we shine UV light on this plate, is it going to glow? No, because what does it not contain? It doesn't contain arabinose. So this plate are transformed cells because they're growing in the presence of ampicillin, right? And they will glow because there is the arabinose to induce and cause that expression of that gene to make the protein. So these guys show no fluorescence, no GFP gene expression. This guy shows the actual expression. These are hopefully what the plates will look like, right? This is under UV light, so you can't really see the growth on this plate and the no growth on this one, but you can kind of see the colonies on this plate, right? But definitely this plate with the arabinose is glowing. I thought I had another slide for some reason. All right.